Hello, I am François Réa. I am the product manager of the NanoSIMS at Camica. So we'll see the NanoSIMS applications in material science part 2, nanotechnologies, and depth profiling. We will start with a study of plasma-driven growth mechanism of metal nanocrystals, published in Nature Materials, mainly by the University of Florida, and the data were recorded, NanoSIMS data were recorded at Pacific Northwest National Lab in Richland, US. So what you see on the left is the small plasmonic seed of gold, and you see some crystal grown after illumination of the growth solution, illumination with light. So with the NanoSIMS, they imaged the gold nanoprism, studying the role of the PVP, the polyvinyl pyrrolidone, in this growth. So the image shown on top has a secondary electron image induced by the primary ions, which were cesium plus 16 kV. We see the gold imaging the nanocrystals, and we see the CN image mapping the PVP. What is very interesting to see is that the CN signal come from the perimeter of the planar twin nanoprisms, suggesting that twin plane defects prompt PVP absorption onto gold nanocrystals. So this is a very nice illustration of the NanoSIMS capability of first mapping a secondary electron, light element like nitrogen together in parallel with gold, heavy elements. So that's one thing. It shows the sensitivity of the NanoSIMS on small objects, nano objects, and also it reveals that it's very interesting to have this normal incidence and normal collection of the NanoSIMS because you have no shadowing. The collection in any direction is very homogeneous because the beam come normal. Finally, we can see the growth of these crystals after illumination. With this input of the role of the PVP in directing the anisotropy growth of the nanoprism, the researcher introduced also iodine in the solution. And on the right side of this slide, you can see the role of iodine in controlling the shape of these nanocrystals in the growth. So you have an overlay of the CN image in green, and the blue represents iodine. So there is a way to control also the shape of the particles depending on the solution. In the coming 3 slides, we will illustrate a simple and very powerful sample preparation to be combined with the NanoSIMS. So it is based on a focused ion beam technique to create and extract a lamella. Here is the schematic of an RF optomodulator, and the cross-section drawing show silicon germanium technology with on each side dopant, so P plus on one side and N plus on the other side. When we see the cross-section, we think that this could be imaged by the NanoSIMS. This was done. So here is the exact drawing that I sent to our lab in the US, equipped with the focused ion beam. So structure of the film, and I asked them to pre-coat them with a thick carbon layer to avoid edges when we will analyze on the NanoSIMS the section. Then I ask them to extract a lamella. No need for a thin lamella, maybe 1 micron thick, it's perfect. No need to have a very low energy cleaning, but then put a few on a diameter of metal. And then we not forget to weld it on the surface for transport. We had some problem before. If you do not fix it on the surface, it can fly. And then this type of cross-section can be directly loaded in the load lock of the NanoSIMS and analyzed directly. And this is what I received by post, 3 FIB samples welded on the top, on the surface of a metallic plate. There are other more sophisticated preparation with a focused ion beam. For example, you can combine with a TEM. So you deposit your lamella on a grid, or other, you can add more sophisticated preparation. But here it is the simplest way to use a focused beam. Here is the image of one cycle acquisition displayed in linear scale on the left and logarithmic scale on the right. And this image is based on a simple FIB technique. It is, you can see, by the way, on the second image, so the elements that are measured here are carbon, oxygen, silicon, phosphorus, and germanium. On the oxygen image, we can see some curtaining due to the high energy fibbing. So this can be refined if one wishes to get better surface conditions for TEM, for example, correlative microscopy. But for here, it's sufficient. So what we observe is that we can image in parallel major elements like oxygen, germanium, together with dopant, despite the fact that on phosphorus it's not so easy. Phosphorus 31 has an interference from silicon 30H. So here the NanoSIMS works at high mass resolving power and resolves this interference without problem, keeping the good transmission. That's the key point. If one wants to detect with a better signal-to-noise dopant, then it is possible to acquire many cycles integrating over some depths of the sample. Finally, for quantitative analysis of such images, one should use reference sample to derive relative sensitivity factor or implanted material, so that we can convert this image of counts into image of atom per cubic centimeter. This example is extracted from a publication from the EPFL group in Lausanne, equal to the NanoSIMS. So it is a study on polycrystalline boron-doped silicon oxide films. These polycrystalline films are used in solar cells as they are transparent. We see on the left a first SEM image of the grain, and we see a strong topography. So for NanoSIMS analysis, first the sample was polished to reduce the topography. Then NanoSIMS images were recorded on a non-doped sample. So we see the zirconium O- and the BO2- ions. So the primary beam was cesium plus at 16 keV. So the zinc oxide is very flat, and the boron image is black, empty. Now let's see the doped sample, and here what is striking is that you see a bimodal distribution of over the zinc oxide grains, you see that the boron is incorporated into one side of each zinc oxide grain. This is attributed to the difference in surface termination between the two sides of each grain. And this will affect the carrier concentration, hence the conductivity. We show an electronic band diagram at the grain boundary, and from this we can, the author, show an example of low resistivity path along the dopant-rich region of the film. So this sort of information taken from NanoSIMS dopant imaging at high resolution helps in developing new film, possibly by controlling the orientation of the grains and improving or optimizing the resistivity for the charge carriers. This sample was brought to us by Professor van der Vorst from IMEC in Belgium. They were suspecting contamination among copper lines polished by CMP, so chemical mechanical polishing. So we analyzed copper lines with the NanoSIMS. We imaged copper lines with the NanoSIMS. We used cesium plus with a beam spot size of 40 nanometer or so. And here you see on the left an image of carbon, oxygen, and then trace element, sulfur, chlorine, and the copper line. And what was very surprising is to observe the sulfur and chlorine just at the center of the copper lines. So this was quite surprising. So here we superimpose copper with sulfur, copper with chlorine, and copper with both. And this was relatively unexpected. But after some study, the plating process used to fill the trenches before the polishing was determined to be responsible for this contamination, trapping the impurities floating on the surface of copper when copper progressively fills the trenches. So again, we see with this example, good resolution, say 40 nanometer, together with the capability to detect light elements at trace level, like sulfur and chlorine. So no other analytical technique could detect this type of contamination. This example shows, again, a trace level measurement. Here it is aluminum that is added as a dopant in copper to optimize its electrical conductivity. We imaged the surface of the film that was provided, selecting oxygen, aluminum, and copper. So note that aluminum is here detected as ALO minus because it was analyzed at this time with cesium. Today, it could be analyzed with O minus and detected as Al plus with similar or better sensitivity. So we zoom on this image to reveal on the left images are displayed in the linear scale and on the right in log scale to reveal the sensitivity of the NanoSIMS. So what we see, the aluminium is segregating at the grain boundaries, and we get with this image very, very clear images in a short time. You see the image is recorded in 10 minutes. So 10 minutes, 15 by 15 micron, 256 by 256 pixels. So it's a fast technique to detect elements at low level with a good resolution. In in the NanoSIMS, the images are recorded as stacks of images, one over the other, and can be displayed by cycle, like here, oxygen, fluorine, phosphorus, or superimposed. So here you see on the left oxygen, fluorine, and at the bottom left aluminium. All these are major elements, and you see phosphorus as a dopant. It can be superimposed. You can directly record and superimpose dopant and major elements. And you can also, if you wish, add many cycles to increase the signal-to-noise ratio and improve the detectability of dopants. Also, with such a stack of images, you can display the image in 3D, combining major elements and dopants. So care must be taken to the original roughness that may create artifacts in the reconstruction, as well as differential sputtering between the different areas of the images. Here is a dopant in a given matrix, silicon, so no problem, and the sample is flat at the surface, so no problem for the reconstruction. The dataset from the previous example can be then rendered in 3D, opened, rotated, coded in different colors for understanding the 3D structure. With a depth resolution of, on the NanoSIMS, 10 to 20 nanometer, and a lateral resolution here in this case, 100 nanometer. So it will be 50 nanometer at best. From this stack, one can also use isosurface rendering to understand better the defined interfaces and shapes. And finally, one can reconstruct depth profiles from small region of interest that can be drawn manually or automatically on the surface. So one can reconstruct depth profile from areas down to, let's say, 1 square micron. The NanoSIMS can be used also as a conventional ion microprobe to produce depth profiling with more than 5 decades of dynamic range. What is specificity? It's the capability to run these analyses from small craters. Here we show arsenic implants, phosphorus and boron shallow implants that are profiled with a good dynamic range, around 5 orders of dynamic range, 5 decades, despite a small crater. So the crater size is 15 by 15 micron, with an electronic gauge of 14% areal, so 5.6 times 5.6 micron. So from this small area, we can reconstruct a quite sensitive depth profile due to the high transmission of the NanoSIMS, despite a quite low beam current, here around 250 picoampere. Here, with this co-standard, arsenic, phosphorus, fluorine in silicon, that we can record from 15 by 15 micron, reasonable profile, quick profiles, that can be run with good dynamic range from with a sputter rate that is, in this case, with a 100 picoamp approximately, we go at around 10 nanometer per minute, so 0.6 micron per hour. So it's not bad for this small area depth profiling. And again, the electronic windows is positioned after the stack acquisition to optimize the dynamic range. Eventually, we can avoid, for example, if there is a particle in the center of the crater, we can reconstruct, move the window and avoid this defect of this particle inside the crater. in this slide, we show that we can use with the NanoSIMS different conditions, primary conditions. We use this a co-implant standard of boron and phosphorus in silicon. And we analyze this with different conditions, cesium plus, BS, Si minus, or O minus primaries detecting positive boron, or O2 minus primary detecting positive boron, or O2 plus primaries detecting BO2 minus. So a choice of condition that allow to optimize, in fact, the parameter you want to push. Whatever the condition we use in this experiment, we have nearly the same detection limit, so around 1e16 atom per cubic centimeter, both for boron and phosphorus. Phosphorus has the classical interference of silicon 30H, but we do not suffer from this with this type of high mass resolving power that we have on the NanoSIMS. So, depending on the condition, the profile will have different characteristics. For example, heavier primary ion, cesium, for example, will give a faster sputtering. Like here, we are at 1 micron per hour. Also, heavier cesium primary or molecular primary, O2 plus or minus, will give better depth resolution compared to lighter single atom, like O minus depth profile. So we can adapt on the NanoSIMS the primary condition to optimize a given parameter. We analyzed this co-implant of phosphorus and boron in silicon with 16 keV cesium in two conditions, 200 picoampere on the left side and 300 picoampere on the right side. So these conditions, together with a crater size of 15 by 15 micron, gave us a sputtering speed of typically more than 1 micron per hour. So this makes the NanoSIMS a productive instrument for small area profiling. And this is achieved despite the fact that we use maybe 1,000 times less beam current compared to conventional SIMS instruments. This is due to the high transmission of the NanoSIMS. Second point, it is important to mention that the NanoSIMS work with a very good vacuum, ultra-high vacuum, so the low minus 10 millibar, and always at high mass resolving power. So no problem for example, the Si30H against phosphorus 31. We remove this interference while keeping a good transmission. And hence, detection limits are in the 1e16 atom per cubic centimeter in these conditions. Traditionally, the NanoSIMS has been used at high energy, 16 keV, in order to get the best lateral resolution. So here we show the depth profile run at 16 keV cesium over 15 by 15 micron. But nowadays, we can also adjust the primary impact energy. And on the right side, we can display the same profile run at 4 keV impact energy over a crater of 10 by 10 micron. In terms of dynamic range, the dynamic range is reduced slightly by less than 1 decade, but still we keep a good sensitivity below 1e17 for both phosphorus and boron. The main impact is on speed. Effectively, on the at 16 keV we go at 1 micron per hour roughly, and at 4 keV in this example we are at 80 nanometer per hour. And this is due to the limitation of the primary beam current by the extraction on the cesium source. This should be improved soon. But still, the conclusion of this slide is that it's now possible on the NanoSIMS to adjust the impact energy to optimize the depth resolution when needed. This example is taken from a publication in Nature Communication from the Stanford University group equipped with the NanoSIMS 50L, where they studied the coating of titanium dioxide nanowires with carbon and tungsten. So you can see the process to coat these tiny nanowires with a mixture of carbon and tungsten. We see the images in SEM and an image of one processed nanowire in TEM, but of course the TEM sees through the nanowire, so thin nanowire, so the beam goes through, and so you do not differentiate the coating from the bulk. So NanoSIMS was used to try to see and visualize this. Trying to make a depth profile from a nanowire is really pushing the small area depth profiling capabilities. So here the crater size was only 1 by 1 micron, and the signal was collected from a central region of interest of a quarter of micron size. Nevertheless, with this small area, a decent depth profile is recorded, you see, with several orders of magnitude. And with standards, one can quantify the tungsten and carbon composition of this depth profile across one single nanowire. So it is possible to acquire such profiles because the sputtering and the collection are normal and the transmission is quite high. It does not require specific and complex sample preparation. Of course, when running depth profile at this small crater size, one must ensure the stability of the sample, so the lab temperature must be stable, otherwise some sample drift correction must be used. Let's conclude. Among the points illustrated in this presentation, so semiconductor, nanotechnology, profiling, we can say that combining with FIB, sample prep, can open to cross-section mapping and permit correlation with transmission electron microscopy. We do not need sophisticated FIB preparation, only a lamella of 1 micron. We can record 1D depth profile, so direct depth profile or reconstructed depth profile from a stack of images. We can record 2D mapping, and 3D image stack can be displayed. The good point and strong point of the NanoSIMS is the compromise sensitivity spatial resolution. This is the strength of the NanoSIMS for material science. So we access sub-ppm sensitivity, so below, let's say 1:16. 1:16 for 15 by 15 micron crater. We can push the crater size toward lower dimension, down to 1-2 micron size, but at the cost of the detection limit, because the volume of material, so the number of ions available, is reducing with the size of the crater. We can adjust the impact energy from between 16 and 4 keV if you want to push the depth resolution, but there will be always a compromise. Higher energy will give you better lateral resolution, down to 50 nanometer. And lower energy will improve the depth resolution by reducing the atomic mixing. So the NanoSIMS can be considered as a bridge, a unique analytical bridge between very high resolution, the atomic resolution techniques like TEM or STEM or atom probe tomography, requiring a sample preparation and limited to a very small field of view. And also between other conventional analysis methods like time-of-flight SIMS, magnetic sector SIMS, or Auger, all lacking whether sensitivity or lateral resolution, or suffering from interferences, for example, quadruple SIMS. Thank you very much for your attention.